lot of people got so seasick they couldn't do anything and just lie down in the cinema because that was the furthest in the center and in the lower part of the ship where it doesn't move so much. But I could go to the bridge and see how we are going down and the waves are coming up. And my name is Heike Zimmermann. I'm a Paleo Ecologist and I'm working with ancient mud. All life contains DNA, and once an organism dies, it settles to the seafloor. And at the seafloor, it can become buried, and the DNA, like this, can be preserved over time. So ancient DNA is like a window to a past that is long gone. And each sample that we have is like a little time capsule. And this time capsule contains the fingerprints of ecosystems from the past. This can be viewed also as a window to our future. So we learn how ecosystems functioned in phases that might have been warmer than today. And this gives us an idea of what we might encounter in the future when climate change is continued to raise our temperatures. With my project, I'm unlocking these little time capsules from Arctic sediments that we've taken from northwest Greenland. I want to find out how far back in time we can use sedimentary ancient DNA in these sediments to understand the connections between climate, the ocean, sea ice, and the biota. We study the Arctic because this is a very important region of climate change. Compared to the rest of the globe, we see that temperatures increase there 4 times more quickly than in the rest of the world. So it is like a frontier for climate change. Two years ago, I went on one of the biggest adventures of my life. I spent 2 months at sea together with more than 100 people on a ship called the GEUS Resolution. And this is not your typical ship. It is actually looking like this. So it has this huge drill tower, which is also called the derrick. And this is where all the pipes go down through the water column, about 2 kilometers through water, and then hundreds of meters down into the seafloor. So we are really anchored during the whole time. During those 2 months at sea, the drilling operations continued 24 hours a day to drill sediments from the seafloor. Once a sediment core is brought up, it comes to something that is called the catwalk, and there the core is getting sectioned into 1.5 meter long core sections. You have to imagine each core is 10 meters long, and it would be really hard to navigate these 10 meters around. And this is where it gets really exciting, because once you open the cores and you see the sediment, you can literally see the ocean change before your eyes. You see differences in colors, you see differences in the properties, like sometimes it's a bit more sandy, sometimes it's a bit more muddy, sometimes you find some shells in it. It is getting really exciting. The GEUS Resolution is essentially a floating lab, so we had all kinds of different labs, and my tiny lab was part of the geochemistry lab, where they had a small microbiology lab. And this was only accessible to me because I had to clean it to ancient DNA standards. I'm dressed this way because I'm subsampling for environmental DNA from the mud line. So the first sediments that come up are the surface of the seafloor, and to prevent contamination from myself, I will wear this. I would use a clean scraper, also with sterile scalpels, to remove the surface of the freshly cut core, because you open and cut the core, and by that you might transfer something of the seawater or the drill fluids down into the core, and you really want to avoid that. I wanted to have a very fresh and clean surface that is not exposed to the ambient air. Ancient DNA is very vulnerable to contamination from modern DNA. I myself have DNA, the environment is full of DNA, and all of this DNA could actually override the ancient DNA signals. And this is because the further down and far back in time we go, the DNA gets more and more fragmented to shorter and shorter fragments. There's more and more damage accumulating over this time. This is why we have to be very careful in the lab and clean excessively. It is actually quite funny because we really clean this lab just to work with mud. Now that we've brought back the samples to Copenhagen, the main method I'm going to use is called metagenomic shotgun sequencing. It's a very interesting method where we sequence everything that is in the sample, like a shotgun. And like this, we can get the DNA signals of all kinds of organisms that have been preserved in these sediments. So you can get DNA from the tiniest of microbes up to the biggest of marine mammals like whales, and all in parallel in one single reaction. Then we need a database to actually make sense of it. We kind of collect all these different taxonomic assignments and count how many times like that codfish was found in a specific sample. Going back over time, we can create time series. And that means we can, with one single method, create hundreds of time series for hundreds of different species. We can look at biodiversity changes over time. So was there more biodiversity in warmer phases versus colder phases? And also how the environment itself changed. We just recently got first results of this project. We have managed to get DNA out of samples up to 600,000 years old. And this is incredible because that means that some of our samples that worked are actually in the top 10 of oldest DNA on the globe. Once I looked into the results and at the species we found, it actually took my breath away. There were all these different species of phytoplankton, zooplankton, worms, clams, even fish. So it is very exciting, and I'm looking forward to getting like the rest of the results soon. Currently, the oldest sedimentary ancient DNA samples are dated to about 2 million years, but we really don't know the limits of how far back in time we can go. And I hope that we can get DNA out of these up to 4 million years old samples to know something about a world beyond those 2 million years.